Okay, so Mendelssohn and Stoff experiments. Just now we have already gone through the detail about it, and then just go through with our answer. You complete this one. Okay, so how they did this experiment? Basically, Mendelssohn and Stoff carried out the experiment by growing the E. coli bacteria in a medium, okay, that contains the heavy isotope. So how in the form of ammonium chloride. Okay, so ammonium here got the nitrogen. So this nitrogen have is a heavy isotope. Okay, so means that the bacteria synthesize their nitrogenous base using this 15N. So means that the DNA will be heavier. So we can subject them to density gradient centrifugation. So using density gradient centrifugation, scientists can separate large molecules such as DNA on the basis of different in terms of their density, but we need to mix it with the cesium chloride, CaCl, okay, and centrifuge at a high speed. So means that you will have the range of the density from the lowest density at the top and the highest density at the bottom, and the DNA molecule will migrate to the regions of the gradients identical to their own density. So what we're going to do here is, after the first round, the team transfer the rest of the bacteria from the 15N to the medium contain the lighter 14N, and lets the bacteria to replicate their DNA. So once they replicate their DNA, no choice in this case because they don't have 15N anymore, so have to use 14N. Okay, they have to use 14N. So by using the UV lights, you will see the DNA band. So this DNA band will show you the presence of 15N or 14N. So the more 15N nitrogen isotope, the denser the DNA. So in density gradient centrifugations, the concentration of cesium chloride is highest at the bottom, darker, of the tubes, and lowest at the top. So DNA molecule move the positions where the density equal to their own, equal to the cesium chloride in which they are centrifuged. So you can see that show the position. If 15N heavy, it will be bottom. 14N will be the top mixture, or we got a hybrid DNA at the center. So when the researchers subject the extracted DNA from E. coli from medium containing 15N isotope to density gradient centrifuge, they accumulated at the high density regions, right? So write down this one actually equivalent to, okay, right here, 15N, 15N. Okay, so this is 0 generation. Okay, 15N, 15N, so 0 generation. So if we have few models here, we have few models, so it means that we need to actually predict the result. Okay, we need to predict the result here. So first DNA replication after transfer to 14N. Yep. So conservative, you're going to have two bands. One band, red color at the bottom. Okay, another one band, blue color at the top. Clear not? Okay, so they are same equal. So this is a 14N, 14N. Then this one is 15N, 15N. Okay, under conservative. Second replications, then you have one thin band. Okay, one thin band, which represent 15N, 15N. And we have blue bands, which is thicker now. Why thicker? Because the ratio is 1 to 3. Okay, ratio 1 to 3. So this is 14N, 14N. So this is a conservative model. Okay. Now, what will happen for the semi-conservatives? If you look at semi-conservative, so we do know that after the first DNA replications, they have one brand at the center only, which represents the hybrid DNA. After the second generations, again, we're going to have the one band at the center. And we have another band at the top. So the one at the top represents 14N, 14N. Another one's, okay, sorry, it's a hybrid DNA, okay, at the center. Dispersive, same thing. We're going to have one band at the center for the first generations, okay, which represents the 29 units of mass, and another one in between. Okay, so this one, 28.5. Okay, clear so far? Okay or not? So now we continue. So this is the results, okay, before transfer to 14 ends. One bands, yeah, as usual, then 1 and 2 bands. So means that only semi-conservative is a correct model. So how we explain? So after transferring the E. coli to 14 end medium, so the DNA isolator after the first DNA replication have a band at the center of the tube. So the findings show the isolated DNA has an intermediate density. So they must be hybrid DNA. So these findings support what? Semi-conservative replication model together with the data dispersive. But the finding inconsistent with the conservative model, which predicted 2 types of double-stranded molecule. Okay, so the result now eliminated the conservative model. But at this moment, are we able to actually differentiate between the dispersive and also semi-conservative? We can't until we continue to the second generation. So after another cycle of DNA replications, the medium with the lighter 14N isotope, 2 bands of the DNA appear in a density gradient. So exactly as predicted by semi-conservative replication model. So DNA band located at the center of the tube consists of hybrid DNA. And then we have the one at the top of the tube contain only DNA with a density of 14N labeled DNA, 14N. So however, the finding now rejected the dispersive model, which predicted that all strands would have a intermediate density. Okay. So the bacterial cells were allowed to undergo the 3rd cycle of DNA replication. So complete the figure 10.13 to show positions of the DNA bands in the tube. So in the 3rd cycle, always remember, okay, 3rd cycle 2 to the power of 3. So 2 to the power of 3 equal to 8. So 2 molecule will be hybrid. Then the remaining 6 molecule will be normal or light DNA. So in terms of the positions, again, you're going to have one band at the center, but Tino represent the hybrid. And then you have one thicker band at the top here, thicker band. Now why thicker? Because the ratio is 1 to 3 now. Okay, thicker band. So this one represents the 14N, 14N, thicker. Okay, why thicker? Because ratio 1 to 3. Are you clear? 2 to 6 means 1 to 3. Hence, calculate the ratio of the hybrid DNA. I want the hybrid DNA to 14N label DNA. 3rd cycle, we know the ratio is 1 to 3. 4th cycle, just now we calculated 1 to 7. Okay, if 5 cycle, how? So if 5 cycle, so 2 to the power of 5, so equal to 32, 2 hybrids, and then 30, that means a light. So answer here 1 to 50. If n cycle, so just now we have showed you guys, so it will be 2 to the power n minus 2, right? 2 to the power n minus 2. This will be after n cycle of DNA replication. So 2 will be hybrid. Always remember, will be 2 hybrid, and the remaining ones will be the 14N labeled DNA. Are you clear so far? Any question? Yes or no? So no question, so then I stop the recording.